Am I audible? Hello? Hello?
Yes, madam. Yes, madam. Please continue.
Am I audible, sir? Hello?
Yes, madam. You are audible.
Hello?
Please start.
Am I audible, sir?
Hello, madam. You are audible.
Okay.
Yes, madam.
Yeah. Thank you, sir. Good afternoon, one and all. On behalf of Oriental College of Pharmacy, I welcome you all on the third day of EFDP. It is a matter of pleasure to know from the feedback of participants that the speakers are highly experienced and PowerPoint is also very clearly visible on YouTube Live. I am also very glad to share with you that Dr. Padma and Dr. Degani has accepted our invitation, and now I welcome Padma Madam. Before that, I will tell Nutan Madam to introduce Padma Madam. Thank you. Over to Nutan.
Yeah, thank you so much, Principal Madam. Good afternoon to one and all. On behalf of Oriental College of Pharmacy, myself, Dr. Nutan Rao, Associate Professor, welcome you all for the third day of 1 week EFTP, Advancement in Pharmaceutical Education. I feel highly privileged to get the opportunity to introduce our first speaker of the day, Professor Padma V. Devarajan. Dr. Padma Devarajan is professor in pharmacy and former head and coordinator MTech Pharmaceutical Biotechnology, Department of Pharmaceutical Sciences and Technology at Institute of Chemical Technology, Mumbai. She is also a member of Board of Governors, President of the Innovation Council, and in charge of the World Bank Technical Education Quality Improvement Program TechWIP at ICT. The Institute of Chemical Technology is the only elite university and center of excellence in state of Maharashtra in India and is among the top institutes in the country and also globally acclaimed. Her research interests include colloidal carriers for targeted delivery in cancer and infectious diseases, veterinary drug delivery, bioenhancement strategies, and mucosal drug delivery systems as alternative to parenteral administration and QBD in drug development. She has over 100 publications and presentations in cited journals and national and international conferences, 5 book chapters in the area of drug delivery. Her book on targeted drug delivery, concepts and strategies, published by Springer, won her the Professor N.R. Kamath Book Award at ICT. Her second book on targeted intracellular delivery by receptor mediated endocytosis has been recently published by Springer. She has filed many patents, international, national, has 7 patents granted and 5 patents licensed and 2 technologies commercialized. Her research is funded through a number of grants from the government and industry, including companies from Japan, Germany, and USA. She is also a consultant to the pharma industry. She was board member on the board of scientific advisors, and chair of the Young Scientist Mentor Proteach Committee of Control Release Society, USA, and is currently chair of the Outstanding Paper Award Committee of Journal Drug Development and Translational Research of Controlled Release Society, USA. She is patron member of Controlled Release Society Indian chapter and member on editorial board of Asian Journal of Pharmaceutical Sciences and elsewhere publication. Professor Devarajan is gold medalist of Mumbai University at BPharm and currently president of Alumni Association of ICT UDCT. She is a nominated fellow of Maharashtra Academy of Sciences, a recipient of the American Association of Indian Pharmaceutical Scientist, Distinguished Educator and Researcher Award 2011, the Vasvik Award for Industrial Research to Women in 2011, and APTI Professor C.J. Shishu Award for Research in Pharmaceutical Sciences. Her publication in the International Journal of Pharmaceutics on gastroretentive drug delivery won the prestigious Udrajit Award in 2015. She won the Bengaluru Nano Innovation Award for a nanosystem developed for veterinary infection, the IPA ACG SciTech Award for Innovation in Solid Dosage Form, and the OPPI Scientist Award 2018. With this short introduction, I now invite and request Professor Padma Devarajan to start with her session. Over to you, madam.
Thank you, Dr. Nutan, and let me begin by thanking Dr. Sudha Rathod and the entire team at Oriental College for giving me this opportunity to discuss some of my work and also some things that we are doing that is a little different than nanosystems. I'll just share my presentation. Just give me a moment. Arshit? Stop screen, Vandal. It is already there.
Already there. Presentation report.
So where do I go? Here? Okay. So it's a little tough because I can't see a single person. But nevertheless, let me talk to you about the topic that is dissolution techniques for nanoformulations, which is a very, very important area and a current area of hectic research. The global formulation market is a huge market of about $600+ million. And in that, the NDDS market is about 13%, with an annual growth rate of 15%. We have 700 NDDS and 1700 technologies. And if you look at this, you will find that the NDDS market is 10 times the Indian pharma market. And in this entire scenario of NDDS, what we have and what we're going to be talking about is nanoformulations or nanomedicine. And nanomedicine, an application of nanotechnology, is used in treatment. It's also used in diagnosis, monitoring, control, etc. So if you look at this particular graph here, you will find that the bulk of nano products are for therapeutics. Then you have the blue, some of them for regenerative medicine, then much smaller amount of work in terms of in vitro diagnostics, in vivo diagnostics, and yes, you also have nano vaccines growing quite rapidly. So the global nanomedicine market, which accounted for 112 billion in 2016, is anticipated to reach 261 billion by 2023, with a compounded annual growth rate of 12.6% right from 2017 to 2023. So currently what we are is in the growth phase of nanomedicine, making it even more critical. Where does nanomedicine really apply? Apart from nano suspensions, which are generally developed for bioenhancement, bulk of nanomedicine is focused on drug targeting based on the magic bullet concept of Paul Ehrlich, the father of targeting, where Paul Ehrlich said that bodies which possessed a particular affinity for a certain organ as a carrier by which to bring the therapeutically active groups to the organ in question. So he was talking of taking the drug with the help of a suitable carrier. So there is an increasing need for the development of such systems, and we are all aware of that, because of certain very critical advantages they provide. One is increased efficacy, coupled with reduced side effects. In fact, with targeting, if I reduce side effects and I don't increase efficacy but just maintain efficacy, that also is considered as a very important achievement. But if I can balance increased efficacy with reduced side effects, that is the ultimate objective of targeted delivery. Reduction in dose or dosing frequency. Again, because of the reduction in side effects, it becomes patient-friendly and highly patient-compliant system. And this is my take on targeted delivery, where I believe that the therapeutic potential of targeted nano delivery systems focus on intracellular infections and cancer, which I personally consider as two very, very major applications of targeted drug delivery. So what do I want to do in targeted drug delivery? I want to deliver drug to the site of action, but at therapeutic levels, and maintain the drug there for sufficient period of time. All of them together are challenges. And with this, I have challenges based on whether I am addressing primary targeting, whether I'm looking at secondary targeting, or whether I'm looking at tertiary targeting. So primary targeting is targeting simply to a particular organ, let me say to the liver, let me say to the brain. But when I talk of secondary targeting, I am talking of delivering to a specific cell type in a specific organ. So if I take this liver as the organ, then whether I'm targeting to the Kupffer cells for a particular infection like say HIV, or whether I'm targeting to hepatocytes for cancer or for hepatitis, this would be an indicator of secondary targeting. And then we can have more advanced targeting. This is tertiary and organelle-based targeting to different organelles in the cell, like the nucleus, the mitochondria, or the lysozyme, or any other, maybe even the endoplasmic reticulum. So to achieve such targeting, what we have is two major approaches. One is passive and the other is active. So in case of passive targeting, what we have is, this is boats which would be bobbing up and down in the water, and they would move in the direction of flow. So it's like you put in a system into the body and the body takes it wherever it wants. And the system cannot decide where it wants to go. That's passive targeting. On the other hand, you can torpedo your system to a particular organ, to a particular cell type, to a particular tumor, and that would be active targeting. But very important for us to understand is whether it's passive targeting or whether it's active targeting, nanomedicine is what is resorted to. So what is the advantage of nanomedicine? Just to recap, is targeted delivery, enhanced efficacy, and decreased systemic toxicity because of decreased exposure to the systemic organs, the systemic tissues, etc. These are two major success stories of nanomedicine. One is Doxil for cancer, and the other is amphotericin B for infectious diseases. So this is what I was trying to tell you, that these are two focus areas which I believe have very high relevance for targeting. And the simple reason is because developing nanomedicine is a huge, huge challenge. And tackling all those challenges and developing nanomedicine for a therapeutic indication that is not very relevant does not make any techno-commercial sense. And hence, when one is talking of nanomedicine or targeted drug delivery, the indication is very critical in terms of choice. Now, what you see here is nanomedicine success stories are further increasing also beyond what I told you, infections and cancer. These are infections, cancer, but you also have for vaccines, you have other kinds of therapy, and then you have analgesics that are developed. And if you look at this, most of the success stories are liposomes, and you have very limited success stories in other systems. Nevertheless, that segment is also growing. But in the current scenario of the pandemic, I think nanomedicine enabled antiviral therapy would probably be the next big revolution in nanomedicine. And we have already started working on this area by developing targeted delivery to the lung by oral administration. We've already done it with tubercular drugs, anti-tubercular drugs, and now we're extrapolating that to antivirals, which could have action against COVID-19, particularly trying to use repurposed drugs. So this could be one of the biggest important areas of current research that would be very, very relevant. So before we look at dissolution of nanocarriers, just a brief peek into the different nanocarriers. Because we need to understand the nanocarrier and then understand how to develop the dissolution technique or how to evaluate in vitro drug release. So normally, if you look at this grid, it says 1 nanometer to 1 micron, that is 1000 nanometer. But in the earlier days, nano typically was limited to 100 nanometer. But if you look at nanomedicine applications, you will see nanogels, nanoparticles, nanocapsules, SLN, liposomes, etc. Everything going beyond the 100 nanometer. So nanomedicine has kind of revolutionized the dimensions of nano and extended it. Of course, nobody goes up to 1 micron, but you do have systems being developed at 500, 600, up to 700 nanometer, which can be safely administered. And then you have polymeric dendrimers, polymeric micelles, then you can have surfactant-based neosomes and microemulsions, polymeric nanoparticles and nanocapsules, then lipidic systems like solid lipid nanoparticles, then liposomes, and then the smaller metal systems like gold particles and carbon nanotubes. So you have a range of nanoparticles and nanocarriers or nanoformulations. So when we look at release, how do we address it? Do we look at it as release for liposomes, release for SLN, or how do we go about it? So these are just pictures of the same thing expanded for you. So this is a nanosphere, a nanocapsule, a dendrimer, polymeric micelles, and this is polymeric lipidic nanoparticles, or what we call as lipomer. And then you have these lipid-based systems. So when you look at all these systems, what is similar in all these systems is not the composition. But what is similar in all these systems is the nanosize. And when developing a release method for such nanosystems, what becomes critical is the size and not the composition. So generally, methods would be common and they would be based on the size of the system, and in a sense, one should be able to use the same method whether I want to evaluate liposomes or whether I want to evaluate SLN or microemulsions. So that is very, very important when one is developing a method. Now, with all these types of systems and large number of liposomes that are marketed, quality control techniques are very important. And particularly for liposomes, where you could have the drug, the hydrophilic drug in the hydrophilic core, or the lipophilic drug in the lipophilic shell, you have the guidance for industry that the US FDA has laid down. And this is the link that I have shown for those who are interested. And you'll be amazed to see what kind of quality parameters they have put down. And industry is really struggling to match these parameters to develop generic liposomal products for doxorubicin, amphotericin B, etc. So it's a huge, huge challenge, and this guidance literally looks at fine, fine details. But very important there is nothing that is mentioned on in vitro release, that they are totally silent on the in vitro release. But currently, there is a lot of work being done by companies in synergy with academics to develop in vitro release methodologies for different nanosystems. And that is what we are going to look at now. Why do I need this in vitro dissolution testing? Because today nanosystems have become very promising. We've seen the market size is growing, the market share is growing. And as on date, there is no standardized in vitro dissolution method available. And when we talk of quality control, release from a system is very critical. So although right now FDA is silent on it, it's going to become one of the important needs as more and more systems are being developed and being approved. So there is an urgent need to develop standard testing methods. While we have 7 apparatus official in the USP, we have testing methods that are specially developed for suppositories, for ophthalmic inserts. So much has happened. Why is it that none of those can be adapted readily for nanosystems? That is because nanomedicine presents very, very special challenges. One is the small size of the nanosystem. We've seen it's less than 1 micron, and like I said, it would generally be in less than 500 nanometer. So there is huge difficulty in separation of nanoparticles from the medium. The systems are very complex. We've seen, we have so many types of systems, which I showed you pictures. The complexity can create problems. Then targeting specific release, and some of them are programmed release, for instance, your temperature sensitive systems or your pH sensitive systems. So this kind of complexity, how does one deal with in an in vitro method or an in vitro setup? And again, for oral, you know what media to use, biorelevant media, but for nanosystems, which are generally injected, what kind of biorelevant dissolution media can you choose? How can you try to mimic the environment-specific release based on pH and temperature? And again, whether there is a need to maintain sink conditions, whether one does not require sink conditions. So these are all various challenges that need to be looked at very carefully when developing the in vitro release method for nanosystems. So let's first look at an ideal dissolution test. It must be reproducible. It must be robust. You must use physiologically relevant media which can give you discriminatory dissolution, which means if I have 2 systems which are supposed to behave different, I must see that difference in vitro also, particularly if I know they're going to behave differently in vivo. It must be convenient and easy to use. It should help you maintain sink conditions. And again, very, very important, adaptable to many formulations. So maybe right from lipidic formulations to polymeric formulations to surfactant-based formulations to maybe even inorganic nanoparticles. This dissolution test that I develop should be formulation independent, and it should be only relevant for any nanosystem, because if one has to develop nano dissolution systems for liposomes separately, nanoparticles separately, the challenge would become too huge and it would be unsurmountable. In vitro dissolution testing methods for nanomedicine is what we will now look at, or rather, what are the methods that have been followed? So you have these categorized under 3 categories: sample and separation methods, membrane diffusion or dialysis sac methods, microdialysis, dynamic dissolution, and 2-stage reverse dialysis methods. So in the sample and separation method, what is done is the nanoparticles or the nanoformulation is directly added to the membrane, to the medium, and separation techniques are applied. And then the drug content in the supernatant or the filtrate is analyzed. So basically, we add the sample to the medium, and then you have to ensure that when I am monitoring the dissolved drug, none of the sample is in that measured sample. So I have ultrafiltration membranes that can be used. So I get a filtrate. I can use ultracentrifugation where I get a supernatant. So very important parameters in the sample and separation methods are this technique that I use, whether it's ultrafiltration, ultracentrifugation, and the agitation conditions, because sometimes the agitation conditions can disturb the nanosystem which we are evaluating. Pressure ultrafiltration is another method where you do an ultrafiltration under pressure. So you can see it's a pressurized filtration assembly through which you filter, and this can happen within 5 minutes. And in such an ultrafiltration method, it is seen that the filter pores do not get clogged. And a simple method is the use of syringe filters. And if this method works, it's normally very simple and very easy to adapt. So these are methods that are suggested to add the sample directly and then separate it out while you are measuring the dissolved drug. But there are challenges in this sample and separation method, and we say separate it out. But this separation is quite a challenge because we need real very high centrifugation and very high energy. Sometimes you need long time and high speed. Like I said, it can destabilize your system. And during the separation process, if it's a long process, drug release can continue and lead to erroneous results. I should stop it. There is an advanced sample and separation method called dynamic dissolution. This utilizes an ion or drug selective electrode, like you have here, to monitor the dissolution release profiles of electroactive drugs. So here you have a reference electrode and a counter electrode. So this is based on ionization or oxidation reduction to generate charged species, and is not suitable for non-electroactive drugs. However, for electroactive drugs, it is a method that is useful without doing separation. The more general methods or the more common methods which are now become very popular and are being investigated very widely are the dialysis methods. Stop that. Nanosystems separated from the release medium through the dialysis membrane that are permeable to the free drug but impermeable to the nanosystem. So my nanosystem would not pass, but my drug would pass through. So this is a schematic. So this is the dialysis sac method. This is a reverse dialysis sac method where the medium is inside the sac and your drug system is outside. And then you have a side-by-side dialysis method where your nanoparticles are inside a glass basket. This looks very much like a USP-1 basket, but this is a glass basket. And you have a dialysis membrane at the bottom through which your drug would, the dissolved drug would come out. So in such systems, the key parameters include the agitation condition, the ratio between donor and acceptor cell volume. So basically, the inside volume should be 6 to 10-fold less than the medium volume. So if I have 1 ml as my sample, I must have minimum 6 to 10 ml of my medium volume, but I may need to have much more if I need to maintain sink conditions, and that would depend on the drug. So for very highly soluble drugs, I can go by this matrix, but if the drugs are not very soluble, I need to use the sink condition matrix to arrive at the medium volume. And then you have the molecular weight cutoff of the membrane, which should not be rate limiting. So the drug that dissolves must freely move out through the dialysis membrane. So a molecular weight cutoff, preferably 10 times more than the drug molecular weight, is what is generally recommended. Sometimes it could be even more than 10. So what are the disadvantages of this dialysis method? One is hindrance to diffusion to the membrane is quite likely, even if we use 10 times more cutoff or even 20 times cutoff, there could be a physical hindrance. Inside the dialysis bag, there is no agitation. And therefore, there could be a challenge of having a gradient inside, and that would again be rate limiting and not be very indicative. That would also cause lot of reproducibility issues. Sink condition, if required, and cannot be maintained in fixed volume dialysis sac methods could be a challenge. In the reverse system, you can cause high dilution of the nanosystem. And one point that is not really put down here, but which I would like to mention is, when I have a dialysis bag, if I go back to this picture and I show it like this, rarely are you going to have a dialysis bag that's going to stand like this. It's going to bend, it's going to fold, it's going to fall to the bottom. So it's not going to have a uniform volume, surface area, etc., and even uniform width or thickness of the system inside. So there are a lot of challenges and lot of causes for a lot of variations that you see. And therefore, progress in this area has moved towards modifying official apparatus. You have the constant volume USP 1 and 2. So this is USP 1 or 2. Here you see this is modified. So you have the shaft, and instead of the basket or the paddle, you will have something else. And more importantly, the continuous flow method USP 4 is also being investigated very aggressively to adapt it for development as an in vitro release system for nanomedicine. So this is a picture of one such modification of USP type 1 by these authors. So here you have an adapter assembly with a dialysis membrane at the bottom. This is called a dialysis cell. So the basket is modified into a dialysis cell with a membrane at the bottom. And this is attached to the USP 1 shaft, USP 1 or 2 shaft, and this would rotate. So this is among a simpler modification. But again, when you have such a huge dialysis membrane and no stirring inside, there would be one more parameter, and that is diffusion gradient. And it is seen that when you have that kind of diffusion gradient, the release can be quite erratic with extensive variation. And therefore, this has been developed differently by this company called Pharma Test, who have developed this adapter called the dispersion releaser. This dispersion releaser is claimed to have high sensitivity for fluctuations in release rate and works well for compounds with poor, moderate, and good solubility. So across the BCS classes is what is claimed. And this dispersion releaser setup, I will show you a better schematic picture so we understand. This is taken from this patent that is filed by the company. And what you see here is this has this dialysis cell, but to prevent that stagnation of this sample inside, this dialysis cell is got a stirring element inside which keeps rotating, and that also serves as a force to move the dissolved drug out of the dialysis membrane. So two purposes served. One is to keep that entire internal system homogeneous and also generate a flux that pushes the dissolved drug. Whatever is dissolved, there is a flux, a kind of centrifugal force, that pushes it through the dialysis bag or dialysis membrane into the dissolution medium that is outside. So this system is claimed to be working very well for nanosystems. The other system is the USP type 4. And this has been used widely for microspheres, which are micron size. But nanoparticulate systems, which have less than 100 nanometer, or let's say even less than 500 nanometer, cannot be studied in USB4 because they are seen to clog the filter, leading to slow flow rates, high pressure buildup in the system also is a challenge that needs to be addressed. And there could also be a situation where the very fine nanoparticles can pass through the filters and actually contaminate the pure drug sample and thereby result in erroneous data. So to overcome these challenges, a novel dialysis adapter was designed to be attached to USB type 4. Now this is a typical USB type 4 or flow through cell. Here you see 7 cells in this apparatus, and this is the flow through adapter, which I will show you. And this is a pump that pumps that this. So you can have what is known as a closed loop and open loop system. Closed loop would be the same medium circulated again and again. Open loop would be the medium just circulated and collected elsewhere, and fresh medium continuously circulated. So if you really have a very, very poorly soluble drug, you can use open loop. If you have soluble drugs, you can use closed loop. So you also have a choice there to maintain sink condition. So this is that adapter. So here you have a dialysis sac and then held together by 2 adapting adapters here. And this is put into your flow through cell, which you see has beads here. And these beads are very critical to ensure uniform laminar flow. So this bead size can be changed, the bead numbers can be changed depending on what is the flow rate one needs to ensure that you get good laminar flow. And this is a closed loop system where there is a piston pump that pumps your medium, collects it into a reservoir, and the same medium is repumped. So this is closed loop. And if it's only collected here and I have another beaker here from where I'm pumping, that would become open loop. So basically, this is where one would monitor the drug release. So we want to ensure that the nano system remains inside here and it doesn't come out, and that is possible with the help of this dialysis adapter. And with the help of this dialysis adapter, since the medium goes along the side, there are also advantages, or rather the disadvantages of filter clogging, etc. are also overcome. So this is a better picture of the dialysis adapter, but a big challenge is to get this adapter because there is only a frame inside and the membrane has to be wrapped around that frame, and then these top and bottom adapters fit. So putting this membrane very uniformly, tightly, without tearing, and then putting these adapters after filling the sample is quite a challenge. But very important, once it's put properly, you can fill 1 ml of sample, you can have different volumes. So you can fill that sample, whatever you want, and you can see this would not change shape. It would not fold or anything. So and it is seen that using this adapter, you get high discriminative power. There is no filter clogging and sink conditions can be maintained. And because it's not very broad, it is considered that the agitation not being inside the adapter is okay. They say it does not matter. Whereas in the pharma test, we saw that they have an agitator there. So this is another view of the same thing. But to overcome the challenge of, the tediousness in handling this adapter, another company has come up with this floatilizer. So this is a dialysis adapter that is ready to use. So all you need to do is fill your sample into this. This is a dialysis tubing which is fit into an adapter, and it is designed such that it fits beautifully into the USB 4 like this. So you just place it in the USB 4 and seal it. So it's a very, very simple system to use, but yes, the challenge is it's quite expensive. Each adapter costs almost 700 to 1,000 rupees. So every test can become quite expensive, but then it's very, very reproducible and very convenient in terms of use. So using this floatilizer, and of course, initially we started with the adapter and then moved on to the floatilizer. Our lab has been doing a lot of work on nanosystems. So I will present to you some of the case studies that we have done with USB apparatus 4 because we are very thankful to Sotax. They generously donated it to us only for use. They took it back. Rather, I must say they loaned it to us for a short period of time. They didn't donate it to us. That's a wrong term. They loaned it to us for a short period of time. And then we could do a lot of studies, and therefore we are very thankful to this. So I will now take you through a few case studies that we have carried out in our lab. So this is inorganic nanoparticles of salmon calcitonin, that is hydroxyapatite nanoparticles. And here we looked at salmon calcitonin, which is a drug of molecular weight about 3000. And our dialysis bag cutoff was about 20,000. So about 6 times. So what we found is the drug comes out very easily. But when we have the nanosystem, the nanoparticles, it comes slowly. So this tells us that the molecular weight cutoff is acceptable because it does not prevent the drug solution from coming out, because within an hour we are getting almost 100% release. And here we are getting significantly slow release, saying that USB4 is an appropriate apparatus for monitoring dissolution. And very important, what is we noticed is there is hardly any variation. We could not really plot standard errors here because the variation was very, very limited for this highly soluble drug. We have studied it for rifampicin, and here what we did is we studied it with dialysis bag placed in USB1 and USB4. So the red line you see is USB1 and the blue line is USB4. We see faster release with USB1, probably because the basket was rotating at 100 RPM. But if you see here, we see erratic variation, very high standard error bars. And if you see this dialysis bag, it doesn't remain long, or even if I say 5 mm, it doesn't have a length of 5 mm when I place it in the basket. It actually just sits in the basket. I would like to use that word, sits in the basket. And when it does that, the surface area, etc. changes. So for each sample, it must have changed, and it's very difficult for you to make it sit the same way. So with USB1 and just a dialysis sac method, the reproducibility was very bad. But yes, if we don't have any other option, we still go by this and see whether, despite the reproducibility, if I can see differences, at least I can rank order my formulations. I may not take that data for any submission, but for my understanding, that may still benefit me, because USB4 is not very easily accessible. But if you look at USB4, you will find that again, the variation was very, very limited in USB4. And the release was slightly lower, but the F2 values were greater than 50, saying that both were comparable because this standard deviations were so high. Now, this is a very, very poorly soluble drug, buparvacone, a drug that you may not be familiar with because it is used for veterinary infections and not really widely used, not used for any human infection. But here what we saw is the SLN release and a suspension of buparvacone. In fact, this buparvacone SLN we have developed by a very innovative in situ nanotechnology, where we just provide a solution of buparvacone. It is diluted in dextrose injection 500 ml to give you buparvacone SLN of about 500 nanometer average size with 98% drug entrapped. Very unbelievable, and this technology is already licensed to industry. Our patent is filed and it is undergoing examination right now. So we were interested in setting up quality profile for that system. So we looked at that. With USB1, we got a release that was lower than here. We got lower drug release. It came down, But basically, in both cases, we had about 40% drug release. Now, this is SLN and this is drug suspension. So, no discrimination between my plain drug and my SLN. But when I go to USP4, I see that my SLN is giving me better release. It's giving me 100% release compared to the drug suspension. And USP4, if you look at the standard error bars of these versus this, you certainly see that deviations are much lower with USP4. So very important finding here is this 100% release, which says that there is likely to be release of the system completely once it is administered. Now, since in this system, USP4, you can, the way you have rotational speeds, here you can manipulate or adjust the flow rates. So we looked at 3 different flow rates, and this was our anti-tubercular formulation comprising 3 drugs, INH, EMB, rifampicin, co-encapsulated in one single step. So in one step, we are able to encapsulate all these 3 drugs in the ratio we would like to put it in. So that is something that the student has really achieved beautifully. And we looked at different flow rates. In this ALF is artificial lysosomal fluid, pH 4.5. And SLF is simulated lung fluid, pH 7.4. So ALF is inside the cell and this is outside the cell in the lung. So basically when you're developing for tuberculosis and delivering to the lung, you could have release outside, but what you want is when the system or the alveolar macrophages take up your particles, there should be release. So what we see is we are seeing increase in release with increase in flow rate, particularly when you go from 8 to 24. So you're 8 to 24, but 16 ml is somewhere in between. It's not really differing either from here or from here. But certainly if you're at 8, it's very low. And when you go to 24, the release rate is going up. And this particular method is also discriminating. It is showing us differences in lysosomal fluid versus the lung fluid, which is also very, very important when one is developing an in vitro release method that needs to be discriminatory. Now, this is the same nanoparticles where we are looking at effect of formulation. So here we have polymer A and polymer B. So increase in polymer B, which is a hydrophilic polymer, results in increase in release rate. And again, what we see is that faster release in artificial lysosomal fluid, particularly here if you see. This is pH 7.4 and pH 4.5. If you see here, there is a significant increase. Even here, there is an increase, but it's not so significant. But here you can significantly see this change, saying that there is good discrimination that one is seeing when you have different media of different pH. So very important is these kind of media could also be considered as biorelevant media. And if I'm able to maintain sync and use such biorelevant media, that would be a very, very ideal kind of condition. For a very drug like amphotericin, very insoluble drug, which is almost considered class 4. If you see here, this is pH 7.4. Here we are looking at 2 different kind of systems. So one is a solid lipid nanoparticle of amphotericin B and the other is a lipid polymeric particle. Just a minute, please. Hello? I have to take the phone. Hello? I thought I am not being heard and somebody from the college is calling. Sorry. So lipomer versus SLN. This is lipid polymeric particle that we have. And this is a lipid particle. So here you see pH 7.4 versus pH 4.8. And here we are seeing very good discrimination. Whereas in pH 7.4, we are not seeing much discrimination. So here, this also tells us that we can actually choose media for discriminatory dissolution. And this method is helping us to do that. And this is a slide that shows USP 1 versus 2 versus 4, combination particles, and this is in simulated lung fluid at pH 4. While you see that the release is roughly higher in USP 2 compared to USP 1, USP 4 it's slightly lower, but what stands out in these three graphs is the low deviation. Is the low deviation. There's hardly any deviation. So in terms of reproducibility, it's very, very good, but this non-reproducibility could be due to the dialysis bag acquiring different shapes, positions, etc., either in the basket or in the bottom of the vessel of the USP paddle apparatus. So what we are doing now, since we are working a lot on nanosystems, I will come to that. So similarly, we are seeing in artificial lung fluid, lysosomal fluid at pH 4.5. See this, the variation is very, very, very low. So before I go to that, all right, we go to the summary. That is, USP 4 with the dialysis cell appears to be a very practical approach, because today you have disposable dialysis cells which are very easy to use. Sink conditions can be maintained, discriminatory dissolution possible, and now using the floatilizer, I can put liposomes, I can put my gold nanoparticle suspension, I can put any kind of system inside that, so it is adaptable to a range of nanoformulations. But very important about USP 4 itself as a dissolution equipment is very expensive, and most institutions don't have it. We have some that are already having it, but many don't have USP 4. So we are working on USP 2. We have developed a very nice adapter with a dialysis membrane, which can be attached, rather used with the USP paddle. And we've already filed the patent, and we're just waiting for some more time before we can disclose what we are trying to do, because we feel that could be one very, very important way of studying nano dissolution or dissolution of nano formulations using USP 2, so that almost anybody could do it. And those adapters would be reusable. Only the dialysis membrane needs to be changed. So again, cost-wise, it's going to be only the membrane cost, which by whatever method you do, you need to bear. So you're not going to spend money on the adapter. So basically, what are we looking at? An ideal dissolution test, which I had characterized at the beginning, should be reproducible, should be robust, should be physiologically relevant, should be convenient. Now with the floatilizer, it is very, very convenient to use. It should be discriminatory. It should enable maintaining sink condition. Yes, when you use the pharma test or what we are working on with apparatus 2, sink condition could be a challenge, and adaptable to many nanoformulations. Very important, because we cannot develop in vitro release separately for liposomes or separately for any other kind of system. It has to be an in vitro release method for the way you say tablets, capsules. Similarly, it would be for nanoformulations across the spectrum. So before I wind up, I would just like to show you the Institute of Chemical Technology. Today it's quite deserted because very few people go there. I mean, some of us keep going once in a while, but not on a regular basis. But more importantly, we have two campuses, one in Bhubaneswar and one in Jalna. Both were opened in 2018. And this year, the third year students would be entering these two campuses. So 2 years almost completed. The second year, with a lot of struggle, is getting completed. But the third year students would soon be entering our two new campuses, not only in Maharashtra, but across the states, that is Bhubaneswar also in Odisha. And this is my very active research group. Some of them have recently passed out during this period. Some have submitted their thesis during this period. And the vibrancy of students is what keeps our lab alive and keeps me ticking. and keeps me young, energetic, innovative, and active continuously. So, a huge thanks to all my students over the years, because research began when I joined as a faculty, and thanks to the students who've been coming in continuously, research has been going on very well. Today, we have a lot of innovations in the lab, lot of technologies that we can credit is what we have developed, lot of innovation awards that have been won by the lab and by students, and all this is thanks to the very vibrant, dynamic research culture of ICT, the freedom and the ambience of ICT, and the team that I've always been fortunate to have at ICT. I would like to acknowledge SOTAX for generous support with the USB4, and like to acknowledge my student Amit Lokhande, who helped me put this presentation together. And a lot of work on USB4 also has been done by Amit, particularly on the anti-tubercular drug particles. And he's a student who's developing this new adapter for nano dissolution, nano formulation dissolution, which can be attached or used with USB apparatus too. And these are two books that our group has done. This one is with Dr. Sayog Jain of NIPER, but this particular book is a total ICT effort, and almost every student of my lab has been authoring this book. And all of us here are from ICT, either currently at ICT or alumni of ICT. So with this, thank you very much for your patient hearing. Thank you.
Thank you so much, ma'am, Devarajan ma'am. It was indeed a pleasure to listen to your talk, and it has definitely increased our knowledge, in-depth knowledge about the dissolution studies, the different apparatus that can be used for the novel drug delivery systems. It was really a treat, especially although I'm not a Sutix person, I'm a chemistry person, but it was really very good and a very thought-provoking session. Thank you so much, ma'am. I'll just see if we have any questions. Um,
Yes.
Many of the participants, they have written that it was a very informative session. It was a very good session. I can see one question from Dr. Harshal Pawar,
Okay.
Hiranand College. He's asking, have you faced any problem in meeting requirement while using modified USB dissolution apparatus?
Today there is no regulatory, no regulations at all. So there is no challenge at all, because whatever you do is what you do. So there are no guidelines for dissolution as on date. That's what I said. Now, if you even look at the liposome guidelines, it does not include in vitro release. It looks at physical and chemical and physicochemical characterization. That's it. It doesn't talk of release. Release is silent because there is no method that is still rugged and reliable and can be accepted across spectrum. So there are a lot of people working on it. So pharma test dispersion releaser, I believe they are working with the USP to try and get it approved, make it compendial. Similarly, I think even SOTAX is trying to get it approved, the USB4. So first, the compendial apparatus has to come. Then the regulations will follow. Now, when I don't have an apparatus, I cannot have a regulation. Because what would be the basis of saying 20% release and 80% release as benchmarks when my method is not standard? So as on today, there is no challenge.
Okay, ma'am. Thank you, ma'am. Again, we are having so many comments. Excellent talk. Very nice session. Nice presentation. Very informative session. There is one participant, Pankaj Sontakke. He has written, thank you very much, madam. Very informative presentation. My question is, what is the standard ideal flow rate for USB4 apparatus and for BCS class 2 and 4, which we should use open or close?
Now for BCS 2 and 4 also, it all depends on what is the solubility and what is the dose. Depending on the dose, you can choose open or close, because the dose will tell you what is the volume you need to maintain sync. So if with 200 ml I can maintain sink, I can go for closed. 500 ml I can maintain. But if I need 2-3 liters, then I will not go for closed, because in that case, it will get too dilute. So then I will go for open and I will continuous flow, and then I will go on monitoring what comes out, in every hour or every 30 minutes or every 2 hours, whatever I want to do. But what was the first part of the question?
My first part was he was asking what is the standard ideal flow rate for USB 4 apparatus.
There is no ideal flow rate as yet. See, for instance, now even if you look at USB 1, what is a pharmacopoeia say? 50 to 150 RPM. So you adjust. Sometimes at 50, you don't get your 85% release. You go to 75, you get it, you choose 75. So similarly here, you have the option of deciding whether you want to be, I have shown you, you can go right from, I think, 4 ml to about 24 or 30 ml. So you can be anywhere in that range, and you have to actually standardize it. Now, once a system comes, let's say Doxil, if a standard method of dissolution is put for Doxil and they say 6 ml flow rate, then when you're testing your system, you will have to go for 6 ml flow rate. Otherwise, as on date, there is no particular flow rate mentioned. It has to be decided based on trial and error.
Okay, ma'am. Thank you once again for elaborating on this particular question. Yeah, ma'am, that's it. Thank you so much once again.
Could I just know how many participants are there?
Ma'am, the number which we can see in live streaming is around 112. 112.
Okay. Okay, 112 participants. Across India or across Maharashtra, or where is it?
Across India.
Throughout the globe.
Pardon?
Throughout the globe, madam.
Throughout globe? Oh, there are people also from outside India. Wonderful.
Yeah.
Nice, very
Hello,
nice.
madam.
Yes.
Do you have any ready thing with you for dissolution of nano emulsions or nano emulsion?
Pardon?
Any ready thing with you for dissolution of?
No, I told you we are working on it. So right now it is in the research and development stage. So right now I don't have anything ready because I'm still in the process of optimizing. But we are certainly working very hard on trying to come up with something that can be attached to or used with USB apparatus too, because almost everybody has USB 1 and 2. So if we come up with something like that, it will be easy for everybody to use. So we are working very aggressively on that.
We will be in touch with you for our research work. Okay, thank you.
Ma'am, there is one more question. It
Okay.
is by Dr. Rajashri Herlekar from Westcop.
Okay.
She is
Okay.
asking, please share some information regarding lipomers.
Okay. Now lipomers are basically lipid polymer hybrid nanoparticles. So you have SLN, which is lipid nanoparticles. You have polymeric nanoparticles, which are 100% polymer. So when we coined this term lipomer, what we did is we had a mixture of lipid, like we took a lot of GMS or stearic acid or polyglycerol distearate, and we took it with a polymer. The polymer we took was gantrase, that is a malic anhydride copolymer, and that we called as a lipomer. So basically now if you want to understand why lipomer and what is the advantage of lipomer over polymeric nanoparticle or lipid, I mean, why did we go for lipomer? So we went for lipomer for a very specific reason, and we were working with a drug called doxycycline hydrochloride, okay, which is for anti-infective, that is used for brucellosis and ehrlichiosis, a lot of veterinary infections which are zoonotic, which come from animals to humans. So it has application for both animals and humans. So doxycycline hydrochloride is so water soluble as a hydrochloride salt, and the base was very difficult to handle. So if we had to entrap the hydrochloride, we needed a polymer. So what we did is we took this maleic coanhydride polymer, which was ionic, so that it could electrostatically or ionically bind to doxycycline hydrochloride. But then when I am looking at an infection and I want to deliver to the RES, I need hydrophobicity. And therefore, I needed something hydrophobic and a lipid. So we said, okay, let's try combining the two. We were not certain whether we would get such particles, but yes, we got particles, beautiful particles, lipid polymeric nanoparticles. So we coined the term lipomer. And very important, what I want to tell you is, while working on this, we even found that with one particular lipid and this particular polymer, we got nanoparticles that had such erratic shapes. They were not round at all. And these particles started going and accumulating in the spleen. So starting with one thing, that trying to entrap a water-soluble drug and ensure that your particles go to RES, we prepared the lipomer. But then we finally found that the one particular lipomer is going to the spleen. We tried to find out why it's going to the spleen. It took us almost 2 years to find out, and then we realized it's because of the shape. The shape was like very, very erratic. It was like any kind of shape like that, odd amoeba shape, let me say, amoeba shape, with its extensions. So that kind of shape was going to the spleen. And then our challenge is, why is this particular composition giving me this kind of shape? So we found that out, and we have a paper also why that is happening. So that research really expanded and unfolded from one thing into why it's going to the spleen. So splenotropic behavior, we explained, that was one good paper that got us huge visibility. I got interviewed by a US magazine called Scientist for that, saying, how did you do it? And then we said, now that we have this funny shape, we must find out why it is funny. So that was done by a third PhD student. And that is a wonderful paper in the International Journal of Pharmaceutics also. In fact, trying to put that paper into getting it approved was a little tough job for us, because convincing somebody about things nobody had seen was quite tough. And we were the first, but we managed. We had 2 rejects, but in the third we managed, and we realized that we were not convincing. So we went on improving our convincing ability, and that really helped us to improve our writing skills also. So there was so much learning, starting with one thing and we've gone to something else, So that lipomer is a very exciting thing, because it is a very useful system to entrap water-soluble drugs in hydrophobic particles. So you have the advantage of good entrapment efficiency, and you have the advantage of the lipidic nature to give you your RES targeting, because to a large extent what we work on is targeting infections. Lot of infections we are looking at, human and veterinary, and of course cancer is also the other big area that we are working on.
It was very nice, ma'am, to know about the story of lipomers, especially because it is from your lab. Very nice, very interesting.
And for me, it's very nice to see a lot of students from ICT at
Yes, ma'am.
Oriental College.
Yeah, nice to see you also, ma'am.
There, Vandana is there,
Yeah.
right?
Yes, ma'am.
Hello, ma'am.
Who else is there? Yes, hello, Vandana.
Yeah.
And who else is there from ICT?
Ganesh
Madam,
Deshmukh,
Ganesh
Dr.
Deshmukh.
Ganesh Deshmukh, and
Oh,
Dr.
okay.
Pradnya Palekar.
Oh, she's also with you.
Yes, yes.
Okay, wonderful. So Dr. Sudha has a very good team.
Yeah.
Ma'am.
And we also have a very good leader.
Very nice. That's a win-win.
Yes, ma'am.
Thank you, ma'am.
Okay.
Thank you, ma'am, once again.
Thank you.
Yes,
Thank you.
thank you so much. Yeah. Okay. Thanks.
Yes, Nutan madam, please continue. Degani ma'am has reached. So please continue.
Okay, fine. Thank you, ma'am. Okay, so it was an excellent SUTIC session. Now the time is to move on to chemistry. And again, this is going to be very interesting. So moving on to the next session, It is again a privileged and a proud moment for me to introduce to you the second resource person for the day, one who is my MPharm and PhD research guide as well. It's none other than Professor Mariam Dekani. I will call her a passionate medicinal chemist and a passionate academician. Let me introduce her. Dr. Mariam Degani is working as a senior faculty at Institute of Chemical Technology, Mumbai since 1998 and currently is Sir Dorabji Tata Professor in Pharmaceutical Chemistry. She has recently completed her term as head of the Department of Pharmaceutical Sciences and Technology from August 2015 to December 2018. Earlier, she was a lecturer at SNDT Women's University, Mumbai from 1985 to 1998. Dr. Degani is working in two broad research areas, that is drug discovery and process chemistry. So this is a very rare combination in itself. In drug discovery, she is involved in computer-assisted rational drug design and synthesis of small focused libraries of potential therapeutic agents. This has resulted in a molecular library of over 600 molecules, which is available for screening on different drug targets. Her drug discovery research involves design and synthesis of small molecules for tuberculosis, Alzheimer's disease, and cancer on several targets, including DHFR, ATP synthase, beta amyloid aggregation, and BCL2 proteins. She has many national and international collaborations to help screen the molecules like Actrec, BARC, TIFR, NCL, Eli Lilly, open source drug discovery, etc. Her other areas of interest include process chemistry with focus on fluorine chemistry, design and synthesis of task-specific ionic liquids, and very recently, flow chemistry-based process optimization. She has successfully completed several projects for industry, mainly in the areas of drug design and synthesis optimization. As an outcome of her extensive research in the above areas, Professor Degani has more than 80 publications in international peer-reviewed journals of repute, including ACS and elsewhere publications, which have received over 700 citations. She has filed two international and several Indian patents. She has also co-authored a book on retrosynthesis as well as written several book chapters and review papers. She is actively involved in various industrial projects and consultancy in the areas of process chemistry and drug discovery. She is a member of several professional bodies and a reviewer for international journals of repute. She has guided 24 PhD and 60 master's students. Currently, there are 5 PhD students and 5 master's students working in her research group. Dr. Degani has been awarded the Distinguished Alumni Award by CU Shah College of Pharmacy in 2007, then Gharda Award for Research Publications in 2009, and Best Teacher Award of ICT 2013 and 2015. She is a fellow of the Maharashtra Academy of Sciences. So, with this short introduction to such an eminent personality, I now invite and request Professor Mariam Degani ma'am to start with her session. Over to you, madam.
Thank you so much, Newton, for that lovely introduction. And at the outset, I would also like to thank all the organizers headed by Dr. Sudha Rathod for inviting me to this session. So what I'm going to do today is share with you some of the ideas that are there in molecular modeling, and this includes some challenges, some success stories, and of course, some other things from our lab itself. So I'm going to start by sharing a few things, and not least, I would also like to share the situation which we are currently having, that is the COVID situation, and how molecular modeling may help in this. So I hope you're able to see the screen now.
Yes, ma'am. It is visible.
Okay, thank you. So let me begin with this little picture. So modeling is not only for beautiful ladies and handsome men, but also for, you can see the benzene ring which is trying to pose over there. So with this, let's see how and where modeling is in today's drug discovery. So modern drug discovery and development process includes a very prominent role of computational modeling. You can see that the computational part, which I'm going to talk about today, starts right at the start of the drug discovery session, where we go on right from target assessment to lead identification and to a certain extent in lead optimization also. So you can see that CAD or CAM as it is called is present right from the beginning of drug discovery to about the place where you get a good molecule, and then pharmacology and other aspects in the drug discovery and development process. So here we have a nice picture of the CAD pipeline. What we can see on the top left-hand part is small molecules. So where do small molecules or fragments come from? They can come from chemical databases. What are chemical databases? They are basically libraries of molecules. There could be public libraries which are available to all of us, or they could be private libraries which can be bought by people when they require. This is one starting point. The other one is the structure of the target receptors. So before we even go to the target receptor structure, we have to identify a therapeutic area, identify the pipeline or the workflow of the therapeutic target, and then go on to the target identification. When these two inputs are there, we can go to the next part. The ligands are finally small molecules, and these, what we draw on paper normally, are just 2D representations. These ligands have to be prepared. By preparation, we mean we convert a 2D structure to a 3D structure, and the 3D structure also has to be something which is conformationally stable. I'll talk a little more about this later. On the other hand, the protein preparation can be done starting from various sources. Could be from X-ray crystallography. It could be from NMR data. And if these are not available, it could be by homology modeling. Again, I'll talk a bit more about homology modeling a little later, but basically what it means is trying to copy another protein which is similar with the amino acids which are present in the molecule, in the protein molecule of interest. So when we have both these aspects, the ligand and the protein, we can go to the next step, which is in the center, that is high throughput virtual screening. In this case, we can have two different aspects. Molecular docking, this means interacting the protein with a ligand to get some data about how well the ligand interacts with the protein. And on the other hand, if the protein structure is not very easily available, or we have a number of ligands which are available, then we can use other methods of screening, that is the ligand-based pharmacophore, including 3D QSAR aspects. The ligand-based pharmacophore modeling can also lead to screening of a large number of molecules. On the other hand, if we do not want any ready-made ligands or molecules, we can do something called de novo or from new, novel molecular design. And we often do that in our lab. Finally, all this could lead to what is called as virtual hits. Virtual hits can be converted to actual synthetic molecules, and in vitro tests can lead finally to compounds which can be taken up further for drug discovery. In the process, we also look at the ADMET properties, and there are several software available for screening some parts of this, especially metabolism and toxicity. However, the distribution of drugs is such a complex process that there is no one model which fits into all the type of ligands which are possible. And not only that, drugs can go into the body and diffuse to different places by passive diffusion. But in addition to that, we have active transport. So the modeling of the distribution part becomes much more difficult than just modeling the target and the enzyme. Another important thing which is done in modeling is binding affinity evaluation. This can be done by molecular dynamics, which is a kind of time-bound picture of how the model drug interacts with the receptor. So on this slide, we see almost the same thing, but in a kind of flow chart. So we start from the left-hand side. So the target disease has within it a metabolic pathway. From the metabolic pathway, the ideal protein, the target protein, which is a druggable protein, is identified. Now, what do you mean by a druggable protein? That means a protein which accepts a drug easily and which is very essential for the functioning of that metabolic pathway. There is no other route which can bypass that protein. So that is important to identify. After validation of all these things, docking can be done, and structure-based virtual screening is done to identify what are called as virtual hits. On the other hand, we see the chemical library. Most of the time, the chemical library is filtered using an ADMET filter or Lipinski's rule of 5. This ensures the screening of only some molecules which are supposed to be drug-like. I'll talk again a little bit about this later on, because these are some of the challenges that you encounter also. Once we come to a virtual hit, either by this way, or if you see the right-hand side, we have another pathway. Known active ligands, that means molecules which are known to act on the receptor, can be converted to a pharmacophoric search, and finally a QSAR model. Machine learning techniques can also be used for this. So you have on this side the ligand-based virtual screening. This can lead to molecules which are then optimized. Once optimization is done in silico, then these molecules can be purchased or synthesized in the laboratory. Experimental validation can be done. And finally, we come to experimental hit compounds. Now, the difference between a hit and a lead is a hit is a compound which passes the initial screening, while a lead passes several other screens, including maybe cell-based assays, toxicity, cell-based toxicity assays, etc. This is the same thing in, again, another way. So I'm not going to go into details on this slide, but for those who are interested, the references at the bottom would give you the details of this. Now let's go to another track. It's interesting to know that since 2000, if you just put the keyword docking as part of a subject or a title of papers, you have more than 6,000 papers in the last few years. So that shows how relevant the word docking is in the discovery of drugs. On the other hand, over here, the thing is slightly different. Here it is not in the title, but in the keywords. So in the keyword molecular dynamics, which I have mentioned in the earlier part, as part of the subject or the title, leads to more than 20,000 hits. The reason being molecular dynamics is a very special technique, and when that is used, there are more instances of people using it in the title or the subject of the paper rather than docking. The reason is docking has become so common now that every drug discovery paper would have some component of docking. But the docking is not just the key part. But when you have a molecular dynamic study, that still remains a key part of the publication usually. Now, if you compare the traditional and virtual screening in terms of expected cost and time requirements, it goes without fail that virtual screening is cost and time effective. However, to translate the virtual screening to actual biological testing may take some time. So what people usually do is do virtual screening at several phases of the drug discovery and therefore save both time and money. Here are some examples of very familiar drugs which have been discovered with CAD. I've just put a few examples over here. That does not mean that others discovered in the last 25 years do not use any of these techniques. In fact, almost all drugs discovered in the last 25 or so years would have used some aspect of molecular modeling at some stage of the discovery. So here we have several antiviral agents, have antibacterial agents, as well as some morphine-like compounds. And we can see that some of them were approved as early as 1995, that is saquinavir, the second compound, which is an inhibitor of HIV and HIV2 proteases. So as early as that, pharmacophore mapping was used to help develop this compound. We can also see the last compound, that is norfloxacin. It's a very old compound, but the class is old, QSAR was used and the compound was approved in 1998. So in the last century, people used more of ligand-based drug design, as the structures of the receptors were not so easy to find 25 years back. But if you see more recently, for example, oxymorphone used 3D molecular docking, dorlosamide used another technique that is called fragment-based screening, and so on and so forth. So this is just a very small glimpse of the number of drugs that were discovered in the last 25 years with the help of some type of CAD input. However, it's not that once you use molecular modeling or CAD-based techniques, it's all done. It's not like that at all. Otherwise, you would have had drugs discovered so fast and so cheaply. No. Why isn't it like that? There are several challenges in CAD. One is CAD needs to integrate with other techniques. That is the wet lab techniques. And not only that, there are so many CAD techniques available all over the world. So many softwares, so many techniques are available, but they don't always talk in the same language. So integration of the techniques with each other, taking the output from one and putting it in the other, still remains sometimes a challenge. The second thing, I had mentioned something about admit prediction. Now over here, I would say absorption by oral route is of course one of the key factors which would be preferable in a drug. And everyone would know about Lipinski's rules in this case, Lipinski's rules of 5. So what happens is, Lipinski's rule of 5 is used as a first filter to remove all unwanted chemicals which do not obey these rules. Is that all right? Not always. The reason being, sometimes antibiotics, for example, do not obey Lipinski's rules, but still are orally absorbed. So Lipinski's rules are just a guideline, but they are not always working. And if we remove some of the molecules right at this stage using Lipinski's rule, we may miss out some very good molecules. Regarding distribution, I had already talked earlier. We have both passive diffusion and active transport. We have several carriers in the blood, including globulins and heme, etc. So it's very difficult to model everything perfectly, and the distribution of the drug is yet to have perfect modeling, but there are lots of inputs here also. Though that's not in the scope of this talk, there are lots of improvements in distribution modeling also. As far as metabolism is concerned, this is also of prime importance because we want a drug to act for a particular amount of time. A very short-acting drug is usually not good, but if you want it just for an eye examination, for example, you would like the drug to be instilled in the eye, work for maybe half an hour, 1 hour, and then be metabolized. On the other hand, some drugs we may want to give as a depot formulation. And in this case, we would like the drug to be metabolized very slowly. Of course, depending on the metabolism, we have excretion and these aspects are also modeled. The T or toxicity prediction is also really important because we do not want to study a toxic compound or a compound which has some toxic moieties in its structure unless we know that it is going to lead to some useful activity. So we have several software which screen out toxic functional groups. However, that is also sometimes not a very good idea because sometimes, as I will show you in the later part of my presentation, a toxic group may be the biologically active moiety of the compound. And in that case, we have to balance the toxicity and the biological activity. Next, we go on to difficult and emerging targets. So not all targets are known. In fact, we may be knowing about 10% of the targets that they are. And these have to be modeled and then can be used for CAD-based drug design. Neglected diseases do not have too many drugs, and these orphan diseases are of special importance, and challenges are there because not so much is known about the targets of these diseases. And therefore, unless those are known, CAD cannot be used. Now we hear about this term chemical space. What does chemical space mean? If we calculate all the molecules that can be made having, say, 50 atoms total, we would end up with so many molecules that it would take some light years for a group of, say, 1,000 chemists to synthesize all of them. Out of this huge chemical space, we are looking at a very small quantum of chemicals. And when we look at ready-made compound libraries, the space becomes even smaller. So if we want to explore new chemical space, then the existing molecules may not be the best starting points. This is a challenge when we are using some of the techniques in CAD, for example, docking, and these challenges can be overcome if we explore chemical space in a different format. Now, multi-target drug discovery and polypharmacology can be a challenge, but it can also be a way forward. For example, in today's day and age, COVID, we are doing drug repurposing most of the time. Why? Because the same drug may be used for a different target. And that also is something which can be explored very well using CAD. However, it is still very challenging to use, and we will see some of this later on. Lastly, CAD requires trained personnel and divulgation, that means divulging the knowledge in form of publications. Of course, in academics, we always want to publish, but many of the drug discoveries by big pharmaceutical companies do not divulge their methods, and that hidden part may not help the future generation. So if that divulging can be done, maybe even after 10 years, it would help a lot in more drug discovery. Those were the broad challenges. There are other things. For example, proteins and ligands are both flexible. It's not like the lock and key mechanism which was told earlier in our, say, 30 years back in our pharmacology session, that the protein is the lock, the ligand is the key, you turn the key and the lock opens. No. Proteins and ligands are both flexible. They are both made up of several bonds which can move, and this makes it difficult to evaluate the energies involved very well. And that goes for the second part also. So protein ligand docking can be done, but has a lot of question marks associated with it. Reason being, if you do not use the correct conformer of a ligand, then you may still get protein ligand docking. But the result may not be the one which you want. The result may not be the one which you will actually see in an X-ray crystallographic structure, or it may not be the one which happens in the body. In that case, you will get several errors. Also, the softwares that were originally made were made specially for small molecules, not so much for peptides. So when peptides have to be docked, then the algorithms that the docking software has to be used, has to be modified before you can get correct results. On the other hand, for example, even in COVID again, there are 3 proteins which come together in order to make the virus reproduce. Can we mimic that in our software? Yes, there are some software, but very sophisticated ones which can do protein-protein docking. Next, we go to the pose versus scoring. So when we do docking studies, we get a score. How well is the ligand bound to the protein? That is called a score. Now, the score is only relevant when the pose is correct. What do you mean by that? The ligand, you can imagine a person is standing, sitting, sleeping, or standing on his hands, and all within a small space, which is the active site of the receptor. Which one is the correct pose? Which one works? That, if that pose is wrong, then the scoring will not have meaning. So that is not so easy to find out unless we have several molecules and we know that all of them work in a certain way. Then we know that if 10 molecules are bound in a certain way and the 11th molecule is very similar, it should bind in that way. However, that is also not always true. On the other hand, what is water solvation? So protein is surrounded by water, and there are very many water molecules which are in the cavities, which are the active sites. When we dock a ligand, do we throw out all these water molecules, or do we keep some? If we keep some, which ones do we keep? How do we know which ones to keep? How do we know which ones to remove? If all this is done manually, that leads to quite a lot of trial and error. However, some sophisticated software do help in showing water solvation. Most ligands bind to receptors, we assume, by non-covalent binding. That means they do not have any covalent bonding. However, it's not always true. For example, molecule like clavulanic acid undergoes covalent binding. It's called a suicide substrate. Can this be mimicked? Again, unless you know the mechanism of action, you cannot mimic this very easily. Also, the algorithms for metals, there are many metals associated with several of the receptors. And the algorithms for metals are not that well known. And therefore, metal binding can be a question mark when we are doing docking. Again, more and more advances are there, and most of these issues are taken care of by the current advances in the software that is available. So we have, once again, several classes of errors and challenges, erroneous assumptions and expectations, data design and content, the choice of the software, conformational sampling, and ligand and target flexibility. I have discussed most of these, so I'm not going to go into the details of all these features, and just mentioned a few of them. I've talked on the first column, that is on the left-hand side, is if you expect that docking is going to give very high affinity compounds, and you're straight away going to go into a best molecule. No, it's not going to happen. I've talked about binding poses, water molecules. I've not talked about one thing, that is allosteric or multiple binding pockets. So now we have to imagine the protein as a kind of sponge where there are holes in the sponge. And there are multiple places or multiple holes where a ligand can bind. If we only explore one binding pocket, then it may not give the correct results. Because allosteric means other than where the protein acts with its inherent ligand or with the substrate in case of an enzyme. So allosteric binding pockets are more difficult to find, and these may be the challenges that we encounter when trying to discover new molecules by virtual screening. Talked about a few of the other things. We go to data design and content. Now over here, just briefly take a few of these. What are decoys? Right at the bottom, you can see inactive compounds which act as decoys are often used to make sure that the protocol that is used is correct. If these so-called inactives are actually having some activity, not unknown activity, then the entire data set becomes wrong. The last point is feature weights. So what part of the molecule is important for the drug activity? Are all the features of the drugs important? Or maybe just one group is really, really essential for activity? If these are not taken into consideration while designing a molecule, then the computational work will lead to erroneous results. So basically, rubbish in, rubbish out. If you don't put the correct stuff in, if the knowledge of the medicinal chemist is not there while adding the structures, then you will not get the correct molecules out. The next column, we have choice of software. Multiple software, as I told you, are available. However, all softwares are not equally good. Tell you a small example right towards the bottom, that is partial charges. For example, a guanidine. A guanidine is structure where 3 nitrogens are around a single carbon. And if this guanidine is charged, where will the charge be? Will it be in the center on the carbon or on which of the 3 nitrogens? If this is not addressed properly by the software, then a lot of other things can go wrong. Just giving you one example over here, but n number of errors happened over here. Another interesting thing which I have personally encountered is molecule preparation. So when a set of molecules is obtained from a chemical library or a data set, it is in a particular file format. Now, what I've actually experienced, of course, this was very long back, years back, was that a biologist was doing this docking. A biologist did not have that much understanding of a chemical structure. So what he imported was structures with a benzene ring, but during the preparation of the molecule, the double bonds were not incorporated. So the benzene became cyclohexane. All structures, there were about 100 structures, all of them looking very funny to me when I just saw them. I said, why are these structures looking so odd? And then I realized that there were no double bonds in any of the structures. And then I realized that it was not the original molecules, but the fact that the molecule was not prepared properly. So by molecule preparation, we mean that we import the structures in a particular file format, and then we add the double bonds, the hydrogens, the tautomers, the charges, the resonance structures, all those. If they are not done properly, there will be a problem. The software can help to a great extent, but it also requires the knowledge of chemistry to be doing this properly. In the last column, we have things which I have already talked about, including conformational coverage, both of the ligand and the target flexibility, and so on and so forth. So I'm not going to go in more details. But all these are definitely to be addressed if we want the correct picture from our virtual screening process. So now going to another tag, some data which I have got from the web. If you see, this is UK data, and in 2015-16, around $100 million was spent on various aspects of CAD. And you can see the graph, though the data was not given to me, has increased almost 8 to 9 fold, and expected to increase, it would increase 8 to 9 fold in the year 2026. So you can see that CAD, both not only the drug discovery part, but even the biological modeling part, that means the genes, etc., is huge today. It's very important. And you can see that most of it is percent is also used in the Asian and African countries. With this background about the various aspects, I have not really covered anything of the techniques because it's impossible to cover, say, about 60 hours of expected work into 1 hour. But I have just given you an overview of the importance of CAD in drug discovery. I would now go on to a few examples from our lab and may not go through all of them. If time permits, I would go through 2 or 3 of them and then come to the conclusion. So I'm going to skip a few slides and go on to, I'm going to start with the modeling itself. So homology modeling is very important when the target structure is not known and you want to make a drug for that target structure. And one example that we had taken, and at that time the structure of Mycobacterium tuberculosis, ATP synthase, was not available. So what was done is, first of all, we have to understand what is the receptor and what it is doing. So I'm not going to go into the details. There are 2 parts of this protein. One is intracellular or rather intramembranous, and the other one is protruding out into the cytoplasm. Now in this part which is protruding out, you have 6 subparts, you can see them, and they are constantly rotating, as can be seen in the middle picture. In this case, we have several subunits. One important amino acid over here is the arginine 186, which has been shown, and the glutamine 61, which is there on the other part of the receptor. When these interact in a particular way, there is movement of the receptor. It literally goes around and in the process converts ADP to ATP by the transfer of a proton. That's it. Now, if we have to stop this from working, somewhere over here, we have to put in a ligand which will stop this interaction of the arginine and the glutamine. So what we had done initially is understood the drug which was already there. At that time, it was not in the market. It was still in clinical trials. So bedaquiline was the drug we had taken. And it was docked with the protein. But you remember that I said the protein structure was not available over there. So what we did is we needed to do a homology modeling to identify and predict a protein structure. Now, what the diagram on the left shows is bedaquiline occupies the receptor very beautifully, interacting maybe with 3 points. But we wanted to design simpler molecules, smaller molecules, without the stereochemistry constraints of bedaquiline. less hydrophobic molecules, because bedaquiline is very hydrophobic, and see if even that works. So that was the purpose of project. And what was done over here initially is we understood the functioning of ATP synthase at the molecular level, understood how it is inhibited by bedaquiline at the molecular level. But before that, we had to have a crystal structure of the protein. Since the crystal structure was not available for Mycobacterium tuberculosis, but a similar protein's crystal structure was available from E. coli. That was our template, our starting point. And by using a software, we use Prime belonging to Schrodinger, and matching the corresponding amino acids, we got a predicted structure of our protein of interest. So we did a detailed sequence analysis, and then from there are lots of details which I have to skip. But what we got finally is a modeling of MTB ATP synthase binding site. So in fact, the protein is really, really very big, and it's not easy to model the whole thing. But what we took is what is circled on the left-hand side, and that is the part which is actually functioning, which we had seen earlier. It's just the one which is going rotating and leading to the ATP synthesis. And that's the part which we modeled. So this is the part, K is the part which is that from the E. coli. And here we have on the right hand side the structure which we could model. This was our starting point, and further we can see a detailed picture over here, and we can see how bedaquiline fitted into this model structure. Fits in very nicely, snugly. Then we kind of dissected the bedaquiline structure and made it suitable for our synthetic capabilities. We kept the quinoline ring, we kept one other aromatic ring, but we modified the rest of the structure, and we designed several molecules. We were not expecting some great nanomolar potency. What we were looking for is simplicity in the structure as compared to the original, and little less hydrophobicity. So here is a list of molecules. You can see that in all these molecules, bedaquiline of course had, which is at the bottom, it had a docking score of minus 9.6. One of our molecules came pretty close, it is minus 9.2. And we can see that pyrrolidinal structures had better scores. By better scores means more negative. More negative means less energy, that means they are more stable. So pyrrolidinal had better scores than morphonyl, and so on. Here you can see morphonyl and pyrrolidinal had vastly different scores. We synthesized these molecules. We also did one more thing. It's a very simple thing, that is to check the 3D shape similarity. So we just superimpose the best conformers of bedaquiline with our molecules to see if there is some similarity. There is a similarity of 0.6%, 0.6, sorry, not percent, which is 60%, 60 to 65%, which is okay. Finally, the proof of the pudding is in the eating, and over here the proof of the pudding is in the testing. So here we see that our MSDPA Q12, the last one, as well as another one, 9, had very good MIC values, the lower the better, but definitely not as good as bedaquiline. We did not have bedaquiline, so we could not test it in our assays. At the same time, they were all non-toxic. If you can see on the right-hand side, the CC50 values are all more than 500. So they are very, very safe. At the same time, they are having good activity. We could understand even post-synthesis why pyrrolidinol fits in better and morpholidin does not fit in well. So when there is a pyrrolidin, there is an interaction of the nitrogen of the pyrrolidin with the GLU L61, which is an important interaction. It is not possible to have that if this pyrrolidin is replaced by a morpholin. So if you see, in case of morpholin, the whole molecule flips. Now remember I had said one of the challenges of docking is the pose. Over here you can see the pose flipping. Is this the real picture, or this is just what we get from docking? Well, what we have realized is the actual activity of the compounds are pretty much in sync with docking. So you can see if you plot this graph, R squared is 0.7, which is quite good for a graph of this type of docking score versus biological activity. Very often we get no correlation. That is one of the drawbacks of docking. But in this particular case, we got a decent correlation between the structures, their docking, and their MIC values when converted in terms of log values. So this, according to us, was a success. We could use this for further improvement of the structures. However, when we add more hydrophobic groups to the molecule docks better but has more hydrophobicity also. So this balance has to be maintained. So as a conclusion of this, we got a novel series of aryl quinoline derivatives, some of which had good anti-TB activity, and we could really correlate. The reason why I've showed this here is because over here, even though we did not have the protein structure straight from crystallography and we had to do a homology modeling, which itself could be questionable, we still got good activity correlationship, which in turn validates the homology model in this case. So, though we have not tested on the enzyme, we have done it on whole cell activity. The correlation did indicate that the compounds that we have made may be inhibiting by MTP ATP synthase. So I would like to know if I have time for one more, or should I go towards the end if the organizers can help me?
Yes, ma'am, you can continue.
I'll continue with one more. Okay.
Yes,
So thank you. Next, I would like to go to one example of molecular dynamics. Now, Molecular dynamics is showing how two molecules interact in a time-dependent fashion. And over here, we have a very interesting example of Alzheimer's, and not going to go into the details of this, but just to tell you that one of the key features of Alzheimer's is the presence of A beta plaques. And A beta plaques are in turn obtained when APP protein is cut at the wrong place to an A beta 40 or A beta 42 fragment. These fragments can in turn form tangles along with rather plaques, and these plaques can in turn form neurofibrillary tangles along with tau inhibitors. So how to avoid this amyloid plaques? This was the question. There was no crystal structure of the A beta plaques because they are very unorganized. However, solution structures from NMR were available, but the exact binding site was not known. So where do molecules bind? And can the binding break up the aggregated molecules was the question. So we did several things. One was proposing a pharmacophore. Second was understanding how known molecules interact with the proposed site. And then finally to the application. To start with, we took whatever was there in literature. There are no therapeutics of this type, so we took diagnostics. And compounds used for beta amyloid imaging were used. In addition, we looked at curcumin because it was known to inhibit amyloid plaques. And resulted in this, we did two things. One was a ligand-based drug design strategy, where we did a pharmacophore mapping and came up with a compound type of scaffold which we should make. This scaffold, of course, ends with fluorine, and the fluorine is used for imaging. So that's another story, but it could be used otherwise also. On the other hand, structure-based drug design was also used, and we started with the amyloid beta protofibril, that is in 2-BEG, which is an NMR solution structure because it's a smaller structure and easier to handle. And what we did is we docked the compounds and first of all found where on the aggregate could the compounds dock. So before we could find, we had to do what is called as a sitemap analysis, where if you see these pictures, there are several places where ligands could dock. Now what this picture is of 5 molecules which are together to form a kind of protofibril. And we can see that docking can either occur at the bottom or at the side. Now, we found that the bottom one was the better place where docking could occur. And we continued with that because all ligands bind better at this site. So whatever ligands we had on hand, we bound to this site. This shows the dock poses for 3 molecules: thioflavin T or THT, curcumin, and fluorbutapir, which is used for diagnosis. And Then what I want to show you is how we studied them over time. So if you see on top, this is from 0 to 1 nanosecond. That fluor beta pair is touching the docking site. However, after 40 to 50 nanoseconds, this molecule is inserted inside the cavity. But the 5 strands do not change much in their structure. So notice that the 5 strands, I'm talking about these 5 strands which you can see from top to bottom, they do not really change so much in the structure. So on the other hand, thioflavin T starts with the same point, but it goes all over. It hovers around the molecule, and it does not really disturb the beta molecule. On the other hand, curcumin again starts binding at the same place as we had seen earlier, that is the site 1. But if you see, after 20 to 30 seconds, the structure of the peptide is considerably different. And if you see after 40 to 50 nanoseconds, it's very changed. What do we conclude from here? Curcumin has the ability to change the aggregation of this and could be used for therapeutics, while the other two, thioflavin and fluor beta, are known to be diagnostic but not therapeutic. So this is the picture we get after molecular dynamics. Dynamics means study of the interaction of the molecules over a period of time. So here we have the final picture. Fluor beta pair inserts itself, thioflavin T hovers around, and curcumin destabilizes the protofibrils. So very interesting study. Requires a lot of computational power, and we took a long time getting these results of 50 nanoseconds. It took several months to get these results. But of course, with a high power hardware, you could get these results much faster. This is showing the RMST of the backbone. In all 3 cases, you can see that curcumin destabilizes the molecules much more, therefore can be used as a therapeutic. And these observations can be used, in fact, have been used now for designing both diagnostic and therapeutics. And very recently, a paper has been published for the therapeutics where part of this work has been used further. So I will not go through this part, but I would like to end by just a small note on COVID. And what I would like to say now is today, first of all, we are in this situation where all of us are mostly sitting at home and interacting with each other because of COVID. And the challenge is to find a solution, to find a repurposed drug for COVID. And over here, we do not have time to do drug discovery starting from scratch. So what people are doing, people are looking at is repurposing of drugs. Does molecular modeling have any scope during this process? Oh, yes. It has a lot of scope, and in fact, there have been plethora of publications in the last 2-3 months on this aspect. There is scope for much more work on this. And over here, in fact, we have written a review paper on this, and you can see that there are a whole lot of targets which we can look. How can we look at them? We can look at any of the approaches that I have listed: docking, de novo drug design, which we have not really done. I'm not really shown in details, but what we did for ATP synthesis, actually de novo, we've designed the molecules from scratch. We have not used docking only. We have used on paper, designed the molecules and then put them on the docking screen. So all these things can be done. However, here we have a time limit. We need to have a drug as soon as possible. And therefore, repurposing seems the best way out. That and perhaps some natural products which are already there. If we can analyze these available compounds against the targets that I have listed here, in fact, more and more targets are coming into the picture, then we can do a good job and find a drug for clinical trials. And if you see, this is a very busy slide, but what I want to show you here is only two parts, the blue part and the green part. The blue part is all the computational approaches. There are so many: signature matching, molecular docking, genetic association, pathway mapping, retrospective clinical analysis, and novel data sources are all based on computational analysis, meta screening, etc. And on the other hand, binding assays and phenotypic screening are the ones which you would use in the wet lab. So there is a lot of scope today and a lot of interest in molecular modeling. And in fact, our government has also launched a hackathon. I hope many of you are aware of it, to find a drug for COVID. You can use many of these targets and repurpose existing drugs by doing molecular modeling as the first step. Of course, that's not the end product. That, if it leads to promising compounds, passing all the challenges which I have described, can be later on tested in vitro and in vivo. So there are lots of challenges here, like too much data is there, big data is there, and access to various types of data is limited at present, especially for clinical trials. But in spite of that, this is a good opportunity for molecular modeling to use in this very critical times of COVID. With that, I would like to just share the pictures of some of the people who have been very active in doing this work at Institute of Chemical Technology, my alma mater, as well as my place of work. Suparna has helped me to put together this presentation, so thanks to her. And here are some more students, including somewhere I can see that some of the students are in the lab, and there's a picture outside the lab too. With this, I would like to thank you all and have a great week, and would like to start if there are any questions.
Thank you so much, ma'am, for such a wonderful presentation. And it is evident from the fact that there are several questions which have been asked by participants. So we are not going to leave you early.
Sure, no problem.
So the first question is from Mr. Shoaib Sayed. He's asking, if the scenario changes and largely the pharma market if it is acquired by herbal formulations, how we can relate it to docking?
It's a good question. So, herbal formulations, when they are done directly, we cannot use docking. But if the ingredients of the herbal formulations are known, as is very often the case, each individual component can be docked and analyzed, and it has been done to a great extent. In fact, I have not presented that part because of lack of time, but there are some herbal extracts of which we have found active components, and we have docked them and found them useful for tuberculosis.
Okay, ma'am. Thank you. Next question is from Dr. Mushtaq Sheikh from Westcop. He's asking, ma'am, how do you see future of CAD in India?
I think it has a great future. But what I think we could do, in addition to using the costly, very, very expensive software, which is made internationally, is we have enough talent. Let us also design our own software. So that, of course, may not only be the medicinal chemist or the computational chemist, but also the software personnel who could be interacting. And in fact, that's part of the hackathon that is there right now. So I see a great scope for this in future, and not only the parts that I have described, but also there are a whole lot of other parts for computer-based information. It may be big data, it may be meta-analysis, it may be a whole lot of other things which definitely require a lot of computational work.
Okay, ma'am. We have one more question from Mushtaq sir. He's asking, ATP is most abundant molecule in the body. Incidentally, many disease problems are around it. What are the challenges and opportunities in this scenario?
ATP is made and used by different organisms, including the humans, of course, in different ways and different approaches. So yes, if we find out the pathways by which ATP is synthesized, by which ATP is converted to ADP and AMP also, then we have a whole lot of possibilities of other drug synthesis.
Okay, ma'am. Then we have one more question from Mr. Firoz Khan, Dr. Firoz Khan. He's asking, sometimes we get good IC50 values for synthesized compounds, but they are not having good binding energy scores for docking study or vice versa. What to do in such situation?
Yeah, that's part of the challenges which I had described earlier. So there are a number of things. IC50, first of all, is in what kind of assays? Do you have wholesale assays, or do you have what type of assays? So if you have enzyme assays, or if you have functional target-based assays, then you would perhaps have a better correlation. However, on the other hand, as I told you, docking depends on a whole lot of things. First of all, the validity of the protein structure. So if you got it even from an X-ray crystal structure, which is today the best source, it still may not be what the protein looks like in solution. And the protein is always in solution in the body. Solution in the sense it's in cytoplasm most of the time, or it's part of a membrane if it's a receptor structure. But it may not be like the crystal structure. That's one challenge. So you have to make sure that the protein structure and cryo-electron microscopy is coming up, maybe the way of the future to solve this issue. As far as the ligand structure is concerned, once again, you have ligands which have multiple single bonds. Multiple single bonds means multiple conformers possible. So for every single flexible bond, if you take a 10-degree conformational change, you have 36 conformers. If you have 5 of these, it would be 5 raised to 36, which is a whole lot. Which of these is the biological active conformer? That is the thing that you have to understand.
Okay, ma'am. There is one more question from Firoz sir. In market, various in silico softwares are available. So, how to determine which software to use for in silico study, or we can use any software and we will get the same result?
No, you will definitely not get the same results with any software. Reason being, the algorithms which go into the making of the software are different. So over here, there's a lot of literature available for comparing software. Don't want to mention any software and bias you for anything, but if you look at literature, you would be able to find that there are some software which consistently give better results than others, and they're not very cheap. So if you can afford, you should go in for the better software, or better still, that's what I'm saying. It's a very, very big effort, but if chemists, biologists, as well as computational people, software people can get together, you can write some good scripting and make a software which is suitable for a particular need. And that's what should be the goal if we want to get software which is reasonable and good. Should not compromise on the quality, but it should be easily available for us also.
Okay, ma'am. Thank you so much. There are certain comments from participants that it was an excellent session. It was very informative session and they have learned a lot. Ma'am, indeed, it was a very inspirational session, especially for medicinal chemists. And definitely all will look into this into details, more details now and with more interest, because the way you have completed the virtual screening and what are the advantages, what are the challenges, what are the disadvantages, and you have taken it so lucidly. So it's definitely going to inspire all the medicinal chemists throughout the globe. So again, I'm very, very thankful on behalf of Oriental College of Pharmacy that you have actually accepted the invitation. Thank you once more, ma'am.
It's really been my pleasure to be on this forum. And once again, thank you all for inviting me to share just a little bit about molecular modeling.
Yeah.
Of course, each aspect of it could go into several lectures and be very interesting to look at that if someone really wants to work on it. Thank you once again.
Thank you, ma'am. Thank you, ma'am.
Thank you, madam. Degani, madam, thank you. Thanks a lot.
Thank
for
you
coming.
and most welcome. Bye.
In time.
Ashish sir, your mic is muted.
Thank you, madam. I think now we are done. And I want to thank all the participants for being present over here And with this, the entire session, the day number 3 is over. The day was full of lots of information from the eminent personalities, and we also have shared the feedback link in the box. And continuously, we are getting messages from our friends, colleagues from all around the country about the quality of the program, and that keeps us inspiring and motivating. So, we would like to thank all the participants, all the delegates who are regularly visiting the site and continuously motivating us. And at the end, I would like to thank our principal madam, all the committee members, anchor for the day, and the speakers who really explained and took the session to a new height, new level. And last but not the least, to the audience for their constant support, which we have been getting. So we want to thank you all. Tomorrow at the same time, 3 o'clock exactly, we will be starting the next session. Stay home, stay safe. Bye.
Okay, thank you.
Thank
Thank you,
you,
sir.
sir. Bye.
See you tomorrow.
Yes, ma'am. Bye.